Hello everyone, today we're going to talk about the innate immune system, and specifically we are going to discuss the complement proteins and the interferon proteins. Complement proteins are proteins found in our plasma. They are produced by our liver. Those complement proteins are definitely a great asset to our immune system, as they are going to assist our immune system in destroying microbes. And we're going to discuss how do they do that. These complement proteins, they are usually inactive, and they do get activated, and we're going to take a look at how do they get activated. And once they do get activated, then they are going to be able to destroy various pathogens. These proteins are usually designated by an uppercase letter C. So instead of saying complement protein 1, complement protein 2, complement protein 3, you get the idea. We just say C1, C2, C3. The numbers, by the way, so when I say C2, C3, what do the numbers represent? The numbers represent the order in which they were discovered. So they've discovered this complement protein, they would give it a number. Later on, they discovered another one, they go ahead and give it another number. So the numbers just represent when they were discovered in order. And usually, as I said, those complement proteins are inactive. Now, when they get activated, we go ahead and we give them name-wise lowercase a's and b's. So, for example, if you have complement protein number 3 and it gets activated, it's going to get activated into complement protein 3A and complement protein 3B, or basically C3B and C3A. Let's take a look at one of the pathways together of how these complement proteins get activated. This pathway right here that we're about to discuss is known as the classical pathway. It's known as the classical pathway because this is the first pathway that they've actually discovered. So let's say that we have a microbe right here with a bunch of antigens on its surface. Now, in our blood's plasma, we do have antibodies, and those antibodies are capable of binding to the antigens on the surface of the microbe if it's a perfect match. What happens is when those antibodies bind to the antigens on the surface of the microbe, we end up with what's known as an antigen-antibody complex. And this complex right here is going to attract one of the complement proteins known as C1, and it's going to activate it. So you can see complement protein C1 goes ahead, kind of binds to that complex right here that we have, and complement protein number 1 gets activated. Now, what's going to happen is once complement protein number 1 gets activated, it's going to activate 2 more complement proteins. And this is something very common about complement proteins. It's kind of like a domino effect. Once one of them gets activated, another one can get activated, etc. So once complement protein 1 gets activated, it's going to activate 2 more complement proteins, complement protein number 2 and 4. Once 2 and 4 are activated, they're going to split, remember how I told you, into A's and B's. So complement protein 2 will split into A and B, and number 4, same thing, into complement protein number 4A and 4B. Now, those two right here, those 2 complement proteins right here, C2A and C4B, they're going to go ahead and activate another one. As I said, it's kind of like a domino effect. They keep activating each other. Once complement protein 3 is activated, as we said earlier, it's going to split into A and B. So as you can see, this is one pathway of how we can activate a bunch of complement proteins. And it all started off with creating that antigen antibody complex. And creating that antigen antibody complex was able to attract one of those complement proteins and activate it. Now, complement protein 3A, it's going to help enhance the inflammatory response. And we're going to talk about that inflammatory response in a bit. Complement protein 3B is going to help destroy the pathogen via two ways. One way is cytolysis, and another one is opsonization. And we're going to talk about those two also in just a few. Let's talk about another pathway of activating complement proteins. So let's say one more time you have a microbe, and that microbe has a bunch of antigens on its surface, or as you can see right here, it has a bunch of lipid carbohydrate complexes on its surface. Now, this time we don't have antibodies. Notice how there aren't any antibodies that are going to bind to the microbe itself. This one right here, it's going to go ahead and in our plasma, we do have complement factors, complement protein factors known as B, D, and P. You see them right here, those cone-like shaped. Those are actually complement proteins, and they are the ones that are going to bind to the microbe. So these complement proteins, they are actually capable of binding to the microbe itself right away, directly. And once they do, they are going to go ahead and activate and combine and like interact right here, as you can see it, with another complement protein known as C3. Once C3 binds and gets activated, it's going to split. And then, as we said, A is going to help with the inflammatory response, and B is going to help with the cytolysis and opsonization. So this is another pathway. If you compare it to the classical pathway, this one right here doesn't involve any antibodies. And you can see right here that certain complement proteins are actually capable of directly making contact with the microbe and activating some other complement proteins. So that's another pathway right there, and they've called it the alternative pathway. This one was discovered after the classical pathway, and therefore they just called it the alternative. There's actually a third pathway of activating some complement proteins, and this is known as the lectin pathway. The lectin pathway was the most recently discovered mechanism for complement activation. And so what happens is we do have some white blood cells in our plasma known as macrophages. And those macrophages, there are phagocytes. What it means is they are going to engulf pathogens and destroy them and digest them within themselves. So when macrophages ingest those pathogens, via phagocytosis, they start releasing a bunch of chemicals known as cytokines. And these cytokines that they release, they stimulate our liver to produce a protein known as lectin. These lectin proteins, they are capable of binding to carbohydrates. And there is a carbohydrate known as mannose. So these lectin proteins, as you can see right here, as I said, they are proteins capable of binding to carbohydrates such as mannose. So we can start calling them MBL, mannose binding lectin. Now, it turns out that these lectin proteins, they are capable of binding to many pathogens. And the reason why they are capable of binding to many pathogens is because they are able to recognize the carbohydrates that includes mannose found on the bacteria surface. So if here is a bacteria and this bacteria has a bunch of antigens and those antigens do contain mannose, then these lectins that were produced by your liver, they are going to be able to bind to it right here. And again, these lectin proteins were produced by our liver due to the macrophages releasing those cytokines as we've discussed. Now, once those lectin proteins bind to the microbe, that's actually going to activate two complement proteins, complement protein number 2 and 4. And once those two go ahead and activate, they are going to split into A's and B's, complement proteins A's and B's. And another thing actually is that not only when those lectin proteins bind to the microbe, they can actually help activate those two complement proteins. Another thing is when this protein binds to the microbe itself, it's going to enhance the phagocytosis of that microbe. So phagocytes that are around, they're going to actually notice that and they're going to come over and they're going to go ahead and destroy that microbe. So you can see you can have two advantages of binding that lectin protein to the microbe. One is phagocytes are going to take notice and they're going to come over and destroy it. And another thing is this complex right here is going to help activate those two complement proteins. And once they are activated, those two complement proteins right here, as I said, it's a domino effect. They're going to go ahead and activate another complement protein, C3, which will split into A and B. So that's a third way of activating some of our complement proteins. And this pathway right here is known as the lectin pathway. Now, we've talked about how to activate those complement proteins, but we didn't talk about once they are activated, how are they going to destroy these microbes? We've mentioned that some of them are going to help with cytolysis. Cytolysis is actually they're going to destroy the microbe by causing the microbe to shatter, to burst. And we're going to talk about how they do that. Opsonization, a way of actually destroying a pathogen is those complement proteins, they're going to attach to the surface of the microbe. And by doing that, they're going to allow the phagocytes to, again, kind of take notice of what's going on and enhance the phagocytosis of that microbe. A third way is they're going to actually help enhance the inflammatory response, and the inflammatory response is also another way of getting rid of pathogens. So let's go ahead and take a look at those three outcomes of complement activation together. So as you saw with all three pathways, the way to activate a complement protein, either by creating an antigen-antibody complex, or certain complement proteins are actually able to directly bind to the pathogen and getting activated this way. In all three pathways, we've talked about how at the end we activate a complement protein known as C3, and once C3 is activated, it splits into C3A and C3B. So let's go ahead and check out together what happens once we get C3A and C3B. How do these complement proteins destroy the pathogens? One way is via cytolysis. Cyte meaning the cell, lysis meaning to destroy. So cytolysis, it's a process of destroying the pathogen, and the way this is accomplished is once we get complement protein 3B, this complement protein is actually going to go ahead and bind or activate complement protein number 5. And once complement protein 5 is activated, it will split into A and B. And you can see right here, complement protein 5B is actually going to activate complement protein 6. And once complement protein 6 is activated, if this is the surface of the microbe right here, notice what's happening. These complement proteins, C5b, 6, 7, 8, and eventually 9 is going to come over. They come and they attach to the surface of the cell, and sequentially, so one comes after the other, and then they all bind together, as you can see right here, like they all huddle together on the surface of the pathogen. And when they bind to the surface of the pathogen, what they're really doing is they're inserting themselves into the plasma membrane of the pathogen cell wall. And when they insert themselves, what they end up doing is they create holes. Or you can think of it as channels. So when they go ahead, and you can see them right here, they huddle together, they keep inserting themselves on the surface of the pathogen, they create these channels. Okay, what's the importance of creating those channels? How is that going to destroy the pathogen? Well, when those channels are created on the surface of the pathogen, the extracellular fluid, so there is fluid outside of our tissues, those extracellular fluids, they're actually going to start entering into the channels. So when all of that fluid starts entering into the pathogen, what's going to end up happening is the pathogen is going to take in like too much fluid and it's going to burst. And that's why this process is called cytolysis. So cytolysis is capable of destroying the pathogens by allowing the pathogen to take in too much water, or basically too much fluid, to the point that the pathogen bursts. And that is possible by basically creating these holes on the surface of the pathogen. Now, which bacteria, if we compare gram-negative versus gram-positive bacteria, which one do you think is going to be more susceptible to getting destroyed by the process known as cytolysis? That would be the gram-negative bacteria. The reason why the gram-negative bacteria are more susceptible to cytolysis is because they have one or very few layers of the peptidoglycan cell wall. And so because they have a very thin peptidoglycan cell wall, they are more susceptible in getting destroyed this way compared to gram-positive bacteria. One more thing to mention about the cytolysis process is when all of these complement proteins that we've discussed earlier attach to the surface of the pathogen, we refer to that as membrane attack complex, or MAC. As I said, gram-positive bacteria, they are going to be less susceptible of getting destroyed via that MAC attack because they have a thicker cell wall compared to the negative gram-negative bacteria. So bacteria that are not capable of being destroyed by that MAC complex, we refer to them as MAC-resistant bacteria. Now, another pathway for those complement proteins to go ahead and destroy pathogens is the process known as opsonization. Opsonization is basically the process of coating the outside of the microbe with one of the complement proteins, and that complement protein is C3b. So once C3 is activated and it splits into A and B, this complement protein goes ahead and starts coating the outside of the microbe, and that is known as opsonization. Once complement protein 3b coats the outside of the microbe, that's going to attract phagocytes. When those phagocytes are attracted to these microbes, what they're going to do is they're going to engulf them and start destroying them. This process right here is known as opsonization, and actually the term opsonization means make it tasty. So if you think about it, those complement proteins are kind of making those microbes more tasty to the phagocytes, allowing the phagocytes to be attracted more to them and therefore engulfing them. So that's another way complement proteins can destroy pathogens, via opsonization. A third pathway is via inflammation. Once complement protein 3 is activated and it splits into A and B, part A is actually going to bind to a cell known as the mast cell, and it binds to specific receptors on the surface of the mast cell. Complement protein 3b actually goes ahead and activates another complement protein known as 5, and it splits into A and B. Part A is also going to bind to the mast cell by binding to a specific receptor. Once those two complement proteins bind to the mast cell goes ahead and starts releasing a chemical known as histamine. Now, histamine is known as it's known to have an effect on our blood vessels. What histamine does to our blood vessels is it dilates our blood vessels. Dilating your blood vessels is actually an important process in getting rid of any pathogens you might have inside of you, because when your blood vessels are dilated, more blood is going to be rushing to that injured scene, for example. So let's say you cut yourself. If you cut yourself, bacteria is going to start entering right away into your tissues. And so you want to go ahead and start your inflammatory response right away. And so one way of actually enhancing that inflammatory response is the complement protein process right here, enhancing that inflammatory response. Your blood vessels, they dilate, and that's going to allow more blood to rush to that site. Another thing, your blood vessels are actually going to become more permeable. Why is increasing the permeability of our blood vessels pretty important? Because that's going to allow these white blood cells, including the phagocytes, if you guys can see right here, the phagocytes within your blood vessels, once your blood vessels are more permeable, that's going to help your phagocytes get out of your blood vessels easier. So they're going to be able to squeeze themselves out of your blood vessels at a much faster pace so that they can get to whatever pathogens is inside of you, and they can go ahead and start launching the attack. That's why if you've ever noticed, when you cut yourself, you do go through that inflammatory response, and that's why it's going to feel pretty hot around the wound, and it might start you might feel that throbbing sensation. And it's going to be red, and that's because histamine is being released by those mast cells, and histamine is dilating your blood vessels and increasing its permeability. So these are three different ways complement proteins, they do help our immune system. They destroy the pathogens. One way is direct. They actually go ahead and create pores on the surface of the pathogen, destroying them. Or in an indirect way, via opsonization, they're not really attacking the microbe themselves, but they are causing the phagocytes to come over and kill the microbe. And then a third way, again, it's kind of an indirect way. Again, they're not killing the microbe directly. They're causing those mast cells to produce histamine, and therefore, when histamine is released and the blood vessels become more permeable and more dilated, again, that's going to cause the phagocytes to come over and destroy these pathogens, or again, other white blood cells can also leak out of the blood vessels, destroying those phagocytes. Complement protein 5A is also known to help attract those phagocytes over, by the way. So these are the three outcomes of the complement activation process. This image right here is a zoomed in image explaining again one more time the inflammatory response that's going to be stimulated by our complement proteins. And we've talked about how complement protein 5A and 3A, they bind to specific receptors on the surface of the mast cells. And once they do, that's going to allow or cause the mast cells to start releasing histamine, and histamine, as I said, is going to enhance that inflammatory response and initiate it by dilating the blood vessels and making the blood vessels more permeable. We also talked about complement protein 5A, how it's actually going to attract phagocytes to the area. So once these phagocytes come to the area, they can actually destroy whatever pathogen is found. Examples of phagocytes, macrophages, neutrophils. We also have some other phagocytes like dendritic cells. And phagocytes, what they do is they engulf those pathogens. So they swallow them up, they start destroying them inside of themselves. Some bacteria out there are actually capable of evading that whole complement protein attack process, and we talked about how, again, those complement proteins, for example, if they're going to attack via the cytolysis pathway, they are actually capable of binding to the pathogen surface and creating those holes and killing the pathogen. Another way is opsonization, where these complement proteins, they coat the surface of the microbe, and therefore attracting the phagocytes to go ahead and kill them. Now, there are some bacteria out there that, for example, have capsules on their surface, and therefore these complement proteins cannot bind to them, and therefore they cannot be destroyed via this way. Your immune system has different ways of killing and attacking those pathogens, but the complement protein in this case won't work against these bacteria. Now, there are actually another set of proteins known as interferon proteins, and there are three types of human interferons, and those are interferon alpha, beta, and gamma. Now, interferon proteins, they are basically cytokines, and they are produced by human cells, and they do have antiviral activity. Or antiviral capability. Now let's talk about interferon alpha and beta first. Interferon alpha and beta, they are going to be actually produced by virus-infected cells. So viruses, they love to hijack our cells, and they inject their nucleic acid within our cells, and they can actually insert their own viral DNA into our DNA. And so they also love to hijack our metabolic processes so that they can use our resources and our enzymes so that they can make their own proteins. And so once a host cell is infected by a virus, some of the cells have the capability of actually producing those proteins known as interferon protein alpha and beta. They produce them, and those proteins, they get released, and they diffuse into uninfected neighboring cells. Once those proteins enter into the neighboring cells that haven't been attacked yet by the virus, they're going to stimulate and induce those uninfected cells to actually start manufacturing messenger RNAs, and those messenger RNAs are going to encode for antiviral proteins. And these proteins that are going to be made are going to disrupt various stages of the viral multiplication that can happen within those cells. So for example, some of the proteins that might be produced is those proteins are going to degrade the viral messenger RNA, and therefore the virus is not going to be able to replicate. Or some of the proteins can actually inhibit protein synthesis. So the virus cannot make any of its own proteins, and therefore the virus is going to die. And so this is one way, and we're gonna take a look at actually an image just in a few, kind of explaining that process right here, the alpha and the beta. But there's another one, there's a third protein known as interferon gamma. And interferon gamma, when it's released, it's actually going to cause the neutrophils and the macrophages to actually come over. So it's going to attract them, and it's going to induce them to start killing the pathogens, such as, like, if there's a bacterial infection, then it's going to enhance those neutrophils and macrophages to come over and start killing those bacteria. So if we take a look at this image together, let's take a look at this cartoon image together right here. So let's say here is a virus. Now the virus is known to inject its nucleic acid into the host cell. Whether that nucleic acid is RNA or if it's a piece of DNA, it's going to go ahead and inject it into the host cell. Once it's injected, it's going to start using the host resources, enzymes, and it's going to start replicating itself. Now, this cell right now is infected. So it's a virus-infected host cell. What this infected host cell is going to start producing is it's going to go ahead and go through transcription and translation to make alpha and beta interferon proteins. Now, once those alpha and beta interferon proteins are made, they're going to be released out of that infected cell. Once they're out, they're actually going to bind to the surface of a neighboring cell, but this cell right here is not infected yet. So it's going to bind to the surface of that uninfected cell, and once it binds to that other cell, it's going to induce transcription and translation of certain genes that are going to encode for antiviral proteins. So that uninfected cell is now going to start making antiviral proteins. Now, those antiviral proteins are not going to stop the virus from actually injecting its nucleic acid into the neighboring cell. Those viruses can still actually go ahead and inject their viral RNA or DNA into that cell. But now what's going to happen is those antiviral proteins are actually going to block the virus from replicating inside of that host. And that is a great thing to do, because if the virus cannot make its own proteins, it cannot use the resources, what's going to happen is that the virus is not going to be able to replicate, and therefore it's not going to infect any other cells, because you are basically inhibiting its replication process. So that's how interferon alpha and beta end up helping out other cells. So those basically proteins, those antiviral proteins, they can work in various ways. They can start degrading the viral messenger RNA. They can start inhibiting the protein synthesis of the viruses, and therefore the virus is not going to be able to replicate, and it's not going to be able to spread to other cells. The concentration of the free iron in our body is actually low, because most of it is actually bound to what's known as the iron binding proteins, such as transferrin, which is found in our blood and in our tissue fluids, lactoferrin, which is found in our saliva, mucus, milk, ferritin in our liver and spleen and red bone marrow, hemoglobin, which is found in our red blood cells. All of these proteins, their job is to bind to the iron. And those proteins are storing and transporting the iron, and therefore you don't see, or you don't find, a lot of just free iron in our body, because they are bound to these proteins. Now, it turns out that pathogens, for them to survive, many of like the pathogenic bacteria that could infect humans, they actually need iron for their survival. And so what they do is they start to secrete proteins. So the bacteria would start producing these proteins known as the cytophores, and those proteins, they're going to start competing to take away the iron from those iron binding proteins that I've just mentioned. That's that's one way they can obtain the iron that they need, is they bind to the iron first by making those cytophores, and they're going to compete and bind to it much more strongly, actually, compared to those proteins. Or one way, they can actually start killing our cells. to get the iron that they need. So for example, Streptococcus pyogenes, this bacteria actually releases hemolysins. Those are proteins that actually destroys and destructs our red blood cells. Why would they want to destroy our red blood cells? Because in our red blood cells, we have tons of hemoglobin, which is bound to iron. So by destroying the red blood cells, they can actually obtain the iron that they need. So either they destroy the cells directly, or they can go ahead and release those proteins known as cytophores, and these proteins will compete with our own proteins, and it will start grabbing on to the iron. There are also antimicrobial peptides. Those are actually short chains of amino acids that are synthesized. And it turns out that these short chains of amino acids, they can actually inhibit cell wall synthesis of the bacteria. So they prevent the bacteria from making their own cell wall, therefore killing the bacteria. They are actually capable of forming pores within the plasma membrane of the bacteria, again, resulting in the lysis and bursting of the bacteria. They're also capable of destroying the DNA and the RNA of some bacteria as well. So they are pretty actually important. An example of an antimicrobial peptide is dermacidin, which is produced by our sweat glands. The fensins, those are another antimicrobial peptides. And as you can see, or as I said, they are pretty important. And it turns out that bacteria are not, up to now at least, we know that they are not capable of resisting them. So those antimicrobial peptides, they can definitely create a lot of damage to these pathogens. And they also found out that some of these antimicrobial peptides, they are also capable of recruiting and attracting mast cells. And we talked already about the mast cells. Those are the ones that are capable of releasing histamine and increasing our blood vessel permeability and dilating our blood vessels. They also, those antimicrobial peptides, they can actually enhance the process of attracting phagocytes, not just mast cells, but they also are capable of attracting phagocytes over, and we know phagocytes are capable of engulfing bacteria and destroying it. So these are another set of proteins that we produce known as the antimicrobial peptides.